The alarming rise in obesity rates, which have more than doubled in the past 3 decades, affecting over 1 billion people worldwide, is a pressing global health concern. This prevalent condition is intricately linked to a host of other metabolic disorders, including type 2 diabetes, cardiovascular diseases, chronic kidney disease, and various cancers. Current treatment options, such as lifestyle interventions, bariatric surgery, and GLP-1 drugs like Ozempic or Wegovy, are not without their challenges. Many patients struggle to access or complete these treatments, and even those who do may find it difficult to maintain their weight loss in the long term. In the quest for more effective solutions, scientists at the Salk Institute have turned their attention to an understudied class of molecules known as microproteins. These tiny proteins, found throughout the body, play crucial roles in both maintaining health and contributing to disease. In a groundbreaking new study, researchers screened thousands of fat cell genes using CRISPR gene editing, uncovering dozens of genes that likely code for microproteins. One of these microproteins, which they confirmed, regulates either fat cell proliferation or lipid accumulation. The findings, published in the Proceedings of the National Academy of Sciences, identify new microproteins that could potentially serve as drug targets for treating obesity and other metabolic disorders. The study also highlights the immense value of CRISPR screening in future microprotein discovery. CRISPR screening is an incredibly powerful tool for identifying important factors in obesity and metabolism that could become therapeutic targets, explains senior author Alan Sagatelian, a professor and holder of the Dr. The more we explore, the more disease-associated microproteins we uncover, and the more potential targets we have for future drug development. When it comes to obesity and metabolic disorders, our energy consumption and expenditure are key factors. Fat cells can grow in both size and number when our energy intake exceeds our energy output, storing excess energy in the form of fatty molecules called lipids. While some excess storage is manageable, too much can lead to fat deposits accumulating around the body, causing whole body inflammation and organ dysfunction. Many complex factors regulate this energy storage system, but the challenge lies in identifying them all and filtering out those that could be promising therapeutic candidates. This has been a long-standing question that Salk scientists have been grappling with for decades. Professor Ronald Evans, an expert on PPAR-gamma, a key regulator of fat cell development and a potent target for treating diabetes, has been working on this issue for years. Several drugs targeting PPAR-gamma have been developed to treat obesity, but they often come with side effects, such as weight gain and bone loss. The ideal PPAR-gamma-based obesity therapeutic remains elusive. When PPAR-gamma drugs fell short, GLP-1 drugs emerged as an alternative. GLP-1, a peptide small enough to be considered a microprotein, regulates blood sugar and appetite. However, GLP-1 drugs also have their limitations, including muscle loss and nausea. Despite these shortcomings, the popularity of GLP-1 drugs highlights the promising potential of microprotein drugs in the field of obesity treatment. Sagatelian's team is now on the hunt for the next microprotein therapeutic, armed with new genetic tools that illuminate the dark world of microproteins. For many years, long stretches of the genome were considered junk and left unexplored. But recent technological advancements have allowed scientists to delve into these dark sections, uncovering a hidden universe of microproteins and expanding protein libraries by 10 to 30%. The Salk team is utilizing innovative CRISPR screening to scour the genome for potential microproteins involved in lipid storage and fat cell biology. This approach accelerates the search for the next generation of PPAR-gamma or GLP-1 drugs. CRISPR screens work by cutting out genes of interest in cells and observing whether the cell thrives or dies without them. This allows scientists to determine the importance and function of specific genes. In this study, the Salk team focused on genes that may code for microproteins involved in fat cell differentiation or proliferation. We wanted to explore if there were any crucial factors we had overlooked in our research on the body's metabolic processes, says first author Victor Pai, a postdoctoral researcher in Sagatelian's lab. CRISPR enables us to identify interesting and functional genes that specifically impact lipid accumulation and fat cell development. Building on a previous study from Sagatelian's lab, which identified thousands of potential microproteins by analyzing microprotein-coding RNA strands from mouse fat tissues. The new study expanded this collection to include additional microproteins identified from a pre-fat cell model. This new model captures the entire differentiation process from a pre-fat cell to a mature fat cell. The researchers then screened this cell model with CRISPR to determine how many of these potential microproteins were involved in fat cell differentiation or proliferation. While we're not the first to use CRISPR for microprotein screening, we are the pioneers in searching for microproteins involved in fat cell proliferation. This is a monumental step forward for metabolism and obesity research, adds Pai. Using their mouse model and CRISPR screening approach, the team identified microproteins that may play a role in fat cell biology. They further narrowed down the list with another experiment, creating a short list of 38 potential microproteins involved in lipid droplet formation during fat cell differentiation, which indicates increasing fat storage. At this stage, all the shortlisted microproteins were still considered potential, because genetic screening identifies genes that may code for microproteins rather than finding the microproteins themselves. While this approach is a valuable workaround for finding these tiny proteins that are otherwise difficult to capture, it also means that further testing is required to confirm their functionality. The Salk team took on this challenge, selecting several shortlisted microproteins for testing. They were able to verify one, which they named adipocyte SMRF1183. Pai hypothesizes that this new microprotein influences lipid droplet formation in fat cells, also known as adipocytes. Verifying adipocyte SMRF1183 is an exciting step towards identifying more microproteins involved in lipid accumulation and fat cell regulation in obesity. It also confirms that CRISPR is an effective tool for finding microproteins involved in fat cell biology, obesity, and metabolism. Research is an ongoing process of improvement, says Sagatelian. We continuously strive to establish better technology and workflows to enhance our discovery capabilities and ultimately improve therapeutic outcomes. Next, the researchers plan to repeat the study using human fat cells. They also hope that their success will inspire others to utilize CRISPR screenings to continue shedding light on the dark world of microproteins, such as adipocyte SMRF1183, which was previously considered unimportant junk DNA. Further validation and screening of new cell libraries will expand the list of potential drug candidates, paving the way for the development of more effective obesity and metabolic disorder therapeutics in the future.